Okay. Whew. All right, we'll start. Do you guys want me to turn like a couple lights on, or is this? Yeah, a couple lights on and a light off. If nobody's here, we're spring break. All right. Okay, that's recording. All right, so we'll continue. I'm going to review a bit of what we did the week before spring break, and we'll continue on from there. How far has Dr. Hasey's gotten along in Chapter 10? Any idea? Is he still in 10A at least? Yeah? Okay. All right. Good. I don't want to get too far ahead. Okay, so we just finished 10A. Okay. All right. Oh yeah, by the way, you guys did really good on exam 2, and oh my god, everybody answered the extra credit. So I had to grade like 300 ones. Usually about half of you decide to skip the extra credit. Apparently this time everyone was really confident with their answers, so it took a long time. Grades are updated, right? You guys see that on Canvas? Okay, good. Yes, question. Everyone attempted, is what I should say. In other words, usually about every exam, only half the students even attempt the extra credit. You guys did really well. Most of you attempted it. I think only like 10 people didn't even bother to try the extra credit. And most of you got like 5, I think. There was a few where, I was kind of lenient. Like if you put one correct thing on there, I gave you 1 point at least, even if everything else was completely wrong. But overall, yeah, you guys did really well. I think the average, I didn't calculate the average, but the average before was like, I think a 76 or something. So plus 5 points extra credit, the average is probably around 80 or something. Yeah.
So you said as long as it was attempted, you got 1 point?
Yes, as long as it was, now if you just wrote like a diagram and the entire diagram is wrong, then I can't even give you 1 point. But yeah, as long as you attempted something, put at least one correct fact on there somewhere, I'd give you 1 point. I was grading all this the last couple days, so, hey, if you believe that the extra credit grade you received was wrong or something, come talk to me and Dr. Hasey's during his office hours. Oh yeah, by the way, you can review, you saw it in the email, you can review your exams starting his very next office hours, which is Friday. Yeah. There's plenty of time this time to review the exam, because exam 3 isn't until like mid-April or something. All right, any questions on anything in general before we start? All right, so, chapter 10, 28, 29, and chapter 30 all have to do with the central dogma of molecular biology. The most important thing that you learn in this class, and probably any biology class, is the central dogma. I'm not going to go over it in detail. You guys should familiarize yourselves with this. Is that the central dogma is based off this diagram here, that everything, life depends on DNA and the expression of genes from DNA. How does the expression of genes from DNA happen? It goes through this process of transcription, where you transcribe DNA to mRNA, and that messenger RNA will get translated into an amino acid sequence, which we learned way back in exam 1, that amino acid sequence will fold into some 3-dimensional shape, which is some functional protein. So all these steps in between and these components here, you have to know a lot of information about. So, the first thing that we talked about last time was the different nitrogenous bases that you have, of which there are 2 types. You have your pyrimidines listed here. I'm not going to go over in detail. And your purines listed here. You do need to know the structures of your nitrogenous bases. But just like with amino acids, come up with little shortcuts to memorize these things. I don't remember every single nitrogen and every single carbon in this thing. Come up with little shortcuts to distinguish one nitrogenous base from another. We talked about certain properties of your nitrogenous bases. For example, and I'm zooming through this because I reviewed it a few times. For example, one property of nitrogenous bases, and therefore DNA and RNA, is the fact that it absorbs UV light. As a reminder, the most important phrase in this diagram is in red right here. And if you want to know what that means, you can look at the YouTube link that I provided last 2 weeks ago. So, skipping. Okay. After nitrogenous bases, we add a layer of complexity to this now. Each of your nitrogenous bases, you can attach a sugar to it. Sometimes you attach a deoxyribose sugar, like so. And let me find here we go. Sometimes you attach a deoxyribose, and sometimes you attach a regular ribose to these sugars. You use the phrase nucleosides to describe these things. I explained how to name nucleosides last time, so I'll leave you guys to study that. After that, we got into some kind of interesting biological facts and applications of nucleosides, which is very often a series of questions on the exam. We talked about one nucleoside derivative called azacitidine, which is a special drug used to treat a particular type of cancer, which, if you want to know the particular type of cancer, is right here, acute myelogenous leukemia. So there's a nucleoside derivative, meaning a drug that looks like a nucleoside, just modified slightly, and just the application of this drug is able to greatly increase the survival rate in patients with acute myelogenous leukemia. The activity, the function that this thing does, let me see if there's a nice diagram for it. Sort of, not really. So a general description of this is DNA, eukaryotic DNA, and human DNA, obviously, is wrapped around a series of proteins called histones. I will go over more detail about this in chapter 28. But DNA is wrapped around histones. And the expression of genes in eukaryotes depends heavily on how DNA is associated to these histone proteins. You can increase the expression of a gene by modifying a histone, and you can decrease the expression of the gene by modifying a histone. I know that's a lot of stuff that hasn't been taught yet in lecture. I will go over it in more detail in chapter 28 and 29. Bottom line, what this drug does is it manipulates and affects the ability for DNA to associate with a particular histone protein. I believe this drug actually, yes, it inhibits a enzyme called DNA methyltransferase. So it inhibits an enzyme whose sole job is to go around methylating histone proteins. What that does is normally, if you let this enzyme go and methylate a bunch of histone proteins in DNA, it decreases the expression of genes. You don't have to know why, but for this particular cancer, the decreased expression of a series of genes causes that acute myelogenous leukemia. Therefore, there is this drug here that inhibits that enzyme. That's one application. Another fact/application regarding nucleoside stuff and chromatin stuff is this interesting fact right here, is the fact that if you look at the DNA methylation pattern of a patient, or of anyone really, the extent to which their DNA is methylated is highly correlated with mortality or lifespan, really. Basically, research is finding out right now, fairly recently at least, that as you age, your chromosomes, your chromatin naturally accrues methylated DNA, which over time leads to various health issues. This accumulation of methylated DNA can be accelerated by certain poor health choices. Smoking, excessive drinking, stress even can also cause an accumulation of DNA methyl groups. So this is research going on right now studying this. That whole field of science that studies the DNA methylation patterns in your chromatin is a term they use called epigenetics. So if you see that term in this class or any other class where they use the phrase epigenetics, they're describing that analysis of DNA methylation. It's technically more complicated than that, but that's the gist of it, is they're studying the patterns of DNA methylation and what health effects arise from that. Continuing on from this, oversimplifying this diagram, all this is showing is that even in development, during development as an embryo develops, there's a massive switch in chromatin patterns in your DNA. Don't worry about exactly understanding everything that is on here. But this switch in chromatin alters the expression of a whole series of genes during development. By switching in chromatin, there's a series of methylations and demethylations. That's all this diagram is trying to tell you, is that chromatin, the nature of chromatin itself, establishes a lot of different things about development and gene expression. This diagram also kind of pairs with that first diagram. It's just telling you that if chromatin changes, transcription factors change also. And I know we haven't gone over transcription factors yet because that's technically chapter 29. So put a bookmark on this statement until we get to chapter 29. More of this stuff, more of that stuff. It's all saying the same thing, that chromatin is modified and gene expression changes, both naturally and in some cases in certain diseases. Other applications. This is another application after we talked about azacitidine. There's another nucleoside-derived drug. I don't know how to pronounce these, but these are the names of the drugs right here. And all they do is that they're specially designed drugs that specifically target an enzyme made by a virus, and specifically the enzyme is thymidine kinase. The cool thing about this drug is that it's so specific for the virus version of the enzyme that it will ignore the human version of the enzyme. And not a lot of drugs are able to do that. A lot of drugs, especially ones with lots of side effects, if you see all those drugs that have lots of side effects, usually end up binding to targets that were unintentional. So this particular drug is able to specifically bind its design, to its designed target to inhibit it, and it's able to ignore human thymidine kinase. Again, I am oversimplifying some of this stuff, and I'm giving you the gist of what these topics are talking about. Another cool application for nucleoside-derived drugs is ICAR, or ACAR, however you pronounce it. Basically, this drug allows you to make mitochondria at a faster rate than normal. It is a performance-enhancing drug. A bunch of people got in trouble for taking this like 5-6 years ago. But basically, bottom line, you can see the one statement they have here in the lab. They give this drug to mice, and they're able to run as fast. Untrained mice are able to run as fast as trained mice when given this drug. And that's because it increases the production of mitochondria. All those applications have to do with nucleosides, nucleoside-derived drugs. Nope. Okay, so after talking about nucleosides, we add one more layer of complexity. We started at nitrogenous bases, then we're at nucleosides, and now we are at nucleotides. Make sure this is recording. Okay, so all nucleotides are very simple. It's just nucleosides with at least one phosphate attached to it. That's it. The name of a nucleotide doesn't change that much. It's the name of a nucleoside, plus you report how many phosphates are attached to it. You guys have known one particular nucleotide since hopefully Bio 1, adenosine triphosphate, right? Adenosine triphosphate is the adenosine nucleoside attached to 3 consecutive phosphate molecules. General functions of nucleotides, it is very important that you know what ATP is generally used for. GTP, CTP, and UTP are each generally used for. GTP we will see again when we get to chapter 30 in specifics. CTP and UTP is saved for Biochem 2 or if you're studying for the MCAT. So, moving right along. Let's see. Okay. So, nitrogenous bases, we added a sugar to it and we got nucleosides. We added a phosphate to it and we got nucleotides. Now when we put the nucleotides together, we get to the really important stuff. We get to DNA, which is a polymer of nucleotides. We get to RNA, which is a polymer of nucleotides. And oh boy, there's a bunch of different types of RNA here, okay? DNA, you guys should know, since Bio 1, DNA is very important. It's where all your genes are, and DNA needs to be as consistent as possible. If DNA fluctuates in terms of pattern, you run into a bunch of mutations, and a whole bunch of issues happen. RNA, because it is a transient kind of temporary version of gene expression, can have a little bit of variability attached to it. Variability in terms of function and variability in terms of patterns. So although there's only one type of DNA, there's a whole bunch of types of RNA. 3 of these you may recognize in previous biology classes, and then there's a couple extra ones here you may not recognize. So the 3 that you should recognize are ribosomal RNA, which is a special type of RNA that is simply that is a structural component of your ribosomes. Hopefully you guys remember what a ribosome does. What is a ribosome's job? Anyone know? What does a ribosome do? Someone's going to have to raise their hand. What does a ribosome do? Yes, go ahead. Protein synthesis, exactly right. So what a ribosome does is it takes part in translation, that part in the central dogma of molecular biology, the very last part. Those are your organelles whose job is to read mRNA and build a protein, build an amino acid sequence. So the ribosome itself, at least a component of it, is made of a special class of RNA called ribosomal RNA. Messenger RNA you should definitely know. We're going to talk about it a lot. In this context, right now, messenger RNA is simply the thing that carries the message from your DNA to your ribosomes. Messenger RNA is transcribed genes from your DNA. And there are ways messenger RNA is modified, depending if you're talking about prokaryotes or eukaryotes. And that's, I think, the very end of 10a and the beginning of 10b goes over that in detail, and we'll spend time on that. Transfer RNA, you won't really get a lot of detail about in this class. At most, they talk about it a lot more in chapter 30, so we'll put a bookmark in that. But all transfer RNA is the RNA that actually carries around your amino acids. During translation, when you're building a protein, when you're building an amino acid sequence, your ribosomes are reading your mRNA to figure out which amino acids need to be brought together, and the amino acids are brought to the ribosome via the transfer RNA subtype. That's covered in detail in chapter 30. All right, those are the 3 core ones. Then there's some interesting ones that are being researched today in a lot of detail. A specific category called siRNA, which technically stands for short interfering RNA, but it's fine if you memorize it as silencing RNA. That kind of is the similar function. But it's involved in gene silencing. I'll explain that when we get to a particular diagram in 10b, what that means. Then there's also other ones other than siRNA, another category called long non-coding RNAs. These are long strands of RNA that don't code for any genes, that don't code for any proteins, but they are still transcribed for some reason. There's research being done right now to figure out, well, what's the purpose of these long non-coding RNAs if they don't actually make any proteins? One function that they have listed here that's being investigated is the fact that these probably they inhibit other mRNAs. Most likely, a particular long non-coding RNA happens to base pair with a particular mRNA and inhibits that mRNA's ability to be translated. I'll draw something when we get to the siRNA stuff, because you'll see it's very similar there. All right. Pictures of a polynucleotide. The one on the left is RNA. And how can you tell it's RNA? Well, you take a look at the sugar. Well, other than the nitrogenous bases, of course. So you take a look at the sugar, and you look at this carbon right here. Let me zoom in. And you look at this carbon right here on the sugar. If this carbon is a OH, it is ribose. It's like regular old ribose. But if you contrast that with the sugars over here, and it is, you notice that there is no longer an OH functional group there. It is deoxyribose. It is lost in oxygen. So the structures on the right represent DNA. This small change in the sugar greatly affects the stability of each of these nucleotides. Way back in Orgo 1 or Orgo 2, you learned that if there's alcohol functional groups, they like to react with everything, right? I don't remember all those SN2 addition, subtraction, multiplication reactions. I don't know. But a particular polynucleotide, if it has a lot of OH groups there, it's going to degrade itself eventually. Contrast that with DNA over here. There is no OH group there to potentially ruin the structure of DNA. So it greatly affects the stability. It's just either the presence of this OH destabilizes the nucleotide and the absence of the OH stabilizes it. So, let's see. This notation here we'll revisit. Let me make sure it's not this. Yeah. This notation here is another way to illustrate a polynucleotide. I will use this notation again later when we get to endonucleases, so we'll skip it for now. But one illustration we can put here is if we're given a particular DNA sequence or RNA sequence, there's a particular orientation we always write it down in, that we always put, there's a particular side we always put on the left side of our page, and there's a side that we always put on the right side of our page. And it is drawn as follows here. The 5' side of a polynucleotide translates to the side with a phosphate at the end of it. The 3' side of a polynucleotide translates to, it's not drawn here, but it's drawn over here. It translates to an OH at the very right side of your nucleotide. So any nucleotide base sequence that is written on any piece of paper, if they don't label which side is 5' or which side is 3', make sure you going from this point on that you know that the left side, whatever the very first base is on the 5' side of that nucleotide sequence. And the very end here, the right side is always going to be the 3' end of your nucleotide sequence. Seems pretty trivial, or what's the importance of this? Trust me, when we do some practice sequencing stuff, it's very important to just start with knowing the direction of a polynucleotide sequence. If you want a biology reason of why you have to memorize this direction, it turns out that if you take a DNA sequence and you transcribe it into mRNA, then you take that mRNA sequence and you translate it into protein, that 5' to 3' direction actually translates to the N-terminus to C-terminus in amino acid sequences. Way back in exam 1, you learned that the very first amino acid is on the N-terminus side, and the very last amino acid is on the C-terminus side. We'll go way back down to the DNA level. That translates to that particular direction. All right. There's a bunch of interesting history stuff, of which none of which you will be asked about on the exam. It is interesting, though. I assume he talked about Rosalind Franklin and his nicknames for Watson and Crick, right? What is it, Rotson and Crook or something like that? It is a pretty interesting story, and stories like this happen all the time in science. People stealing credit and getting famous for it, and some no-name in some lab somewhere ends up losing out on it. This is one of the rare stories where people found out about it. For the exam, you won't be asked anything about this, but if you're interested on learning the story of Rosalind Franklin, there's plenty of links on here for you to follow. Let's see, blah, 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 blah. Okay. So, we talked about nitrogenous bases, nucleotides. We started putting together the nucleotides to form DNA and RNA. But now we got to look at the three-dimensional structure of DNA. We put two strands of DNA together. What shape does it form? What patterns are there? Some numbers are listed here. Really, the only important information here is the fact that DNA is in a shape called an anti-parallel double helix, which really, if I go back to this picture here, you should know by now that DNA is a double helix shape. It's this weird twisting ladder shape. What they mean by anti-parallel is it has to do with this direction in DNA again. All right, I told you that in an individual DNA sequence, we write it out from 5' to 3' because the 5' end represents the phosphate end, and the 3' end represents the very last OH in a nucleotide. But if you pair two different nucleotide sequences together, you find that they have to match each other in an anti-parallel orientation, where one, in this illustration, 5' is on top, 3' is on bottom, and the other one over here, 5' is on bottom and 3' is on top. This just so happens to be the reason for this is because of some complicated organic chemistry orbital reason that I don't care about and you don't need to know about. But the end result is the fact that they pair with each other like so in this anti-parallel arrangement. So, blah, blah, blah. I believe this figure illustrates this. One critical component, or two critical components about DNA base pairing is the fact that you need to memorize that A's always bond with T's, and G's always bond with C's. That should be easy. That's not too difficult to memorize. The second fact is kind of interesting. Is the fact that for every AT bond, they are only connected to each other via 2 hydrogen bonds, just 2 weak hydrogen bonds. So all DNA in every organism is held together via weak intermolecular forces. It is hydrogen bonds, but it is technically a weak intermolecular force. There are no covalent bonds connecting these 2 strands of DNA together. Each strand individually, like if I look at just the left strand, it's connected by covalent phosphodiester bonds. But when I put the strands together, they are only connected to each other via hydrogen bonds. A's and T's are connected to each other via 2 hydrogen bonds. Don't worry about the 0.28 or whatever. While G's and C's, interestingly enough, are connected via 3 hydrogen bonds. This subtle difference, it doesn't sound like a big deal, but this subtle difference, if you carry this out over an entire genome, you have regions of DNA with lots of A's and T's in them, meaning those regions of DNA will be easier to open up. And to contrast that, you will have regions of DNA in your genome that have a bunch of G's and C's all in a row. Those regions of DNA will be a lot harder to open up because there's more hydrogen bonds keeping them together. Again, it's not a big deal, but this is one of the small differences that cause all this complexity that you see in life. All the differences in gene expression from one organism to another, in part, is because just the number of hydrogen bonds keeping these things together. Okay, moving right along. Again, don't worry about memorizing length of genomes of organisms. Okay, so now we get into more detail about mRNA. And this is now, from this point on, they're going to go with specific details talking about prokaryotes, and they're going to contrast these details with eukaryotes. They do this in this chapter, they do it in chapter 28 and 29 and 30. A majority of questions that you'll be asked about in this class will have to do with the prokaryotic system of something. Prokaryotic mRNA, prokaryotic DNA replication, prokaryotic transcription, prokaryotic translation. When you are asked something about eukaryotic versions of those things, it is most often going to be asked to you in a way where they just want you to contrast the eukaryotic thing with the prokaryotic thing that you learn in detail. I will give you examples as we go through it. So let me make sure I don't skip anything first. Okay, so I'm going to try to draw. Let's see. Okay. So first, prokaryotic. All right. So the very end of 10A and the beginning of 10B is talking about mRNA. It turns out mRNA is, other than just talking about genes, that mRNA itself is quite different from prokaryotes and eukaryotes. When they talk about prokaryotes, they sometimes use the phrase polycistronic mRNA or polycistronic genes, something like that. The key phrase is polycistronic. I first memorized this word as being as associated with prokaryotes because they both start with the letter P. But what polycistronic means is that if I look at one single mRNA strand, here's a 5' end and here's a 3' end, and I'm not going to write in all the bases, but imagine we're zoomed out a lot on this mRNA strand, but if we zoom in a bunch, we're going to see in here written a bunch of bases, okay? This mRNA strand, when we use the phrase polycistronic, what it means is on one mRNA strand, we can transcribe multiple genes. Some base pair representing gene A would be here. Right next to that, there's another base sequence pattern representing gene B. So the genes, remember, these are the things that are eventually going to be translated into proteins. Gene A is going to be made into protein A. Gene B is going to be made into protein B, and so on and so on. I'm going to contrast polycistronic mRNA with the other version of mRNA, but let me conceptualize this for you first. So again, the phrase polycistronic mRNA means that on one mRNA strand, you can have multiple genes transcribed onto it. Okay, on every one mRNA strand, you can have multiple genes, and they are right next to each other. Another word they use to describe it when genes are right next to each other is the phrase contiguous. So you might see that phrase show up either here or on the practice exam or on the worksheets. But whenever you see the phrase contiguous, they are describing polycistronic mRNA and prokaryotic mRNA. That's because these genes are right next to each other. There's very small, and sometimes there might not even be any gaps in between these things. Okay. All right, so this is polycistronic mRNA, that one mRNA gives you multiple genes. It might help to really understand this if we contrast this with the other type of mRNA, and you might see the phrase monocistronic mRNA. Okay. So what is monocistronic mRNA? This is found in eukaryotes. And monocistronic mRNA is for every one mRNA, you will only get one gene that is transcribed. Okay? You will get one gene that is transcribed. So this entire thing, it's not to scale or anything. This entire thing is the transcribed version of gene A. They read a particular gene in DNA in your eukaryote, and when they transcribed it, it came out to this one mRNA strand containing only that gene and nothing else. Okay? The reason why that is when you look at genes in eukaryotes, they are not contiguous. An overwhelming majority, probably 99.9% of genes in eukaryotes are not contiguous, meaning there's significant empty gaps between genes in your DNA sequence. Meaning one gene, one base pattern in your genome codes for gene A, then there's this huge gap of bases that don't really do anything, and then you reach the second gene in your eukaryotic genome. So because of this gap between genes in your eukaryote, when they decide to transcribe genes in eukaryotes, they can only do it one at a time, one gene at a time. Okay. So this comes with some complications. In prokaryotes, because you're able to transcribe one gene after the other really easily, you just transcribe one single mRNA strand and you get a bunch of genes all in one go. You don't really have to modify your mRNA at all for it to be translated. There's some details in chapter 29 and 30 that I won't go over yet, but each one of these, there's a start site and an end site with each of these, and they're all right next to each other. Because of that, we use the phrase, or similar phrases, that there are no introns in prokaryotes. And because of that, we don't really use the phrase exons either. It's a minor notation thing. Bottom line, I'll explain, is that there are no introns in prokaryotes. What that means is there's no gaps in your coding sequences, that everything that is transcribed from the start sequence to the end sequence will be translated into a protein. You contrast that with eukaryotes, and you might remember from Bio 1 or 2 that in eukaryotic genes, there are things called introns and exons. Okay, so in prokaryotes, we don't have that. We don't have to worry about introns or exons. In prokaryotes, it's just every gene that you see is going to be translated. In eukaryotes, we have a slight complication in that after you transcribe a gene, it's actually in a pre-mRNA form. It's in a pre-mRNA form, an unfinished mRNA form. This is only in eukaryotes. In prokaryotes, when you transcribe it, you're done. Yes?
What are those arrows pointing to on the polycistronic?
Start and end sites. Yeah, somewhere in there is a start site, somewhere in there is an end site. Don't worry about it. It's just to illustrate that these are very close to each other, while in eukaryotes you don't have something like that. So, another synonym, by the way, for pre-mRNA, in case you see this written somewhere in your notes, is hMRNA, or HN it might be, but bottom line what this means is heterogeneous RNA, which means RNA that still contains, I'm going to draw an example of pre-mRNA, that still contains introns and exons. So what are those things? I will explain in a second. Okay, so again, we've zoomed out a lot. This is still an mRNA sequence, and I just haven't drawn it, but there are bases written in here. This region of base patterns represents an exon, and right next to it is a region of bases representing an intron. This whole thing is still one gene. This whole large structure here is still one gene. It just includes both exons and introns. What these are, exons are the important stuff. Exons actually code for amino acid sequences. Okay, these things are actually the parts that you need, that your ribosomes need to read in order to make a functional protein, a functional amino acid sequence. Introns, I am oversimplifying it, but at this point you can think that introns are just junk. Okay, just for the purposes of this illustration, if you go into really technical, introns are very important, but just for this illustration, they don't code for anything. They do not code for amino acid sequences. So, prokaryotes, we didn't have to worry about this. Prokaryotes, when we transcribe this thing, we can immediately translate it into proteins and we're happy. Over here, after we transcribe it, we get this pre-mRNA that still has a bunch of useless stuff in the middle of it. So a feature of eukaryotic transcription is a thing called splicing. And this, I will actually go to the diagram they have in your notes, and I believe it's like the last slide in 10A, is this thing here. Okay? This green-red thing represents a eukaryotic pre-mRNA that was made in transcription. The green stuff represents introns, the red stuff represents exons. Because the green stuff doesn't matter, in eukaryotes there's a series of enzymes and a structure called a spliceosome that goes in and removes these introns and splices together the exons. In eukaryotes, a unique feature of this splicing mechanism is that you can splice it in many different patterns here. One single pre-mRNA, as you can see in this illustration, you can see two different patterns that you can splice the exons together. And if you arrange them in different orders, you get two different proteins. Okay? So although one mRNA encodes only one gene in eukaryotes, that one gene, depending on how you splice it together, can make several different proteins. So that's where complexity arises in eukaryotes, is because of this alternative splicing here. Okay, so key feature again in eukaryotic transcription, eukaryotic mRNA, is that it's initially made in this pre-immature heterogeneous form. You remove the introns and you splice together the exons, and you can make a bunch of different types of proteins from a single gene depending on how you splice them together. It is more complicated than that, but this is good enough for an illustration at this stage. Let me flip to 10b to make sure I didn't forget anything regarding this. Okay, so before I move on, does anyone want me to explain those components again? It is a little weird if you haven't seen it before, but yes. Mm-hmm. What's that? I can't hear you. You're saying re-explain the previous thing? Yeah, okay. So prokaryotes do not have this issue. Prokaryotes, you have just one mRNA strand, has a bunch of genes on it, it gets transcribed, and there's no problem. We use the phrase polycistronic to describe that. Eukaryotes, one mRNA strand is only able to contain one gene, but eukaryotes have this extra thing that happens after transcription, where that gene, that pre-mRNA, they remove the useless parts of it. They remove the introns. They remove the green stuff on this diagram. And the important stuff, the exons, the stuff that actually codes for amino acid sequences, they splice them together. They stitch them together in various different patterns. And whatever pattern it stitches them together in translates into a different protein. I believe there's another diagram that may illustrate this right here. Yep, all right, so let me explain this one. So this diagram is a re-explanation of polycistronic mRNA, okay? And how do I know this is polycistronic mRNA? Well, let's take a look at the genome. Here's gene A, and right next to gene A is gene B. There is no empty space between these two things. And right next to gene B is gene C. It turns out that this DNA fragment in prokaryotes, when it is transcribed, you make a single mRNA strand that has the mRNA corresponding for gene A, right next to it the mRNA corresponding to gene B, and this one, the mRNA corresponding to gene C. Another difference, I should say, between prokaryotes and eukaryotes is something you might remember in Bio 1. Remember that prokaryotes do not have nuclei, or do not have a nucleus organelle, right? What that means is that DNA in prokaryotes is just floating around the cell, and that when you transcribe DNA in a prokaryote and you get this mRNA, you can immediately translate it because there is no nucleus in prokaryotes. When you transcribe the DNA into mRNA, there's a ribosome waiting right by it to begin translating it into protein. This diagram tries to illustrate that for you, that it turns out that this end of mRNA, this left side here, was transcribed a long time ago. It was transcribed earlier in this diagram. And although the very back end is still being transcribed, take a look at this. The ribosomes right here are translating the mRNA, the very front end of the mRNA. So a feature of prokaryotes is that transcription and translation can happen simultaneously. One end of your mRNA that was finished being transcribed is currently being translated into protein. In prokaryotes, you do not have to wait for transcription to finish. If you have an mRNA, even if it is still being made, you can begin translating it. And the reason why that is there's no nucleus separating your DNA from your ribosomes. Okay? We can contrast that, I believe. They might not actually have a picture of this. Contrast that with eukaryotes. We know that eukaryotes have a nucleus, and that DNA in eukaryotes is stored in a nucleus, but ribosomes are out in the cytosol. So in eukaryotes, you have to actually transport your mRNA out of the nucleus to the ribosomes and have it be translated. So we talked about one way eukaryotes modify mRNA in eukaryotes is this splicing thing. There are two other things that is done in eukaryotes. One is attaching this thing you see on the end here, a poly-A tail. This is done to stabilize the mRNA. Why is that important? Well, in eukaryotes, we have to transport the mRNA all the way from the nucleus out of the nucleus and to ribosomes. The mRNA might not make it the whole way. So what happens in eukaryotes is we attach a poly-A tail to it to stabilize it. Prokaryotes don't do this. They don't need to do this. A second thing that is done, and it's not illustrated here, a second thing that is done to stabilize eukaryotic mRNA is that you attach what's called a 5' G cap. You don't have to know the really specific details of that. It's just another thing to stabilize eukaryotic mRNA. And we'll go over some practice questions of this at the next SI session, but I'll stop here for now. Yes, question?
So the caps are kind of like running from the nucleotides? Yes.
And whoever has the sign-in sheet, if you could bring it up here.